This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 36, recorded on January 31st, 2012. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
Also known as D-Cubed.
Right.
Am I taking that too far, Dickson? Does it bother you?
No.
How you doing, Dickson?
I'm doing fine. Very good.
Should we wrap this up?
Sure. Are there any questions?
The last day of January.
Yeah.
I'm always sad when January passes. Do you know why?
Yeah, because it's tax time soon.
No, because January has my birthday in it.
Oh, yes.
And it's a self-serving emotion, I understand.
Of course.
One must take care of oneself, no?
I think that's true. Yep.
All right.
But not at the expense of others.
No, I wouldn't do that. Nevertheless, do me a favor and push the play and record at the same time buttons on that. Play and record. Those two at the same time. Let's see what happens. Is play green?
Play hasn't gone on.
Yeah,
play
on,
is green.
on.
No,
okay,
it's on.
Good. That's my backup recorder in case something happens here.
Right.
I will, of course, remove that.
Yeah.
All right, Dickson.
Yes.
The other night I was reading through my paper folder.
Good.
Which is not actually made of paper.
No.
It's a folder on the computer full of papers that are interesting.
Does it sound like paper when they open it?
No,
Do they make that little noise on the screen?
No.
Because when you crush up something, it does that.
No, I don't think computers should imitate life. I mean, do you like when you flip a page on a computer, it has to look like a page?
It looks like a page.
I don't see what that's called skeuomorphic. I don't see why you have to do that. Anyway, I found this paper in Science.
Yes.
Exploitation of the intestinal microflora by the parasitic nematode Trichuris muris.
Yes, I read the same paper, by the way.
I'm glad we're on the same page.
We're on the same pages.
And we did discuss Trichuris trichiura.
We did.
A while ago.
Vince, that's very good of you to remember.
Do you remember?
That. I do, of course I do.
In fact, I'm going to dig up the episode.
Right.
On microbeworld.org.
Right.
/
Right.
What is it? / Dickson. It's supposed to roll off your tongue.
Yeah, right.
TWiP.
TWiP. Oh, TWiP. Of course, we TWiPed the Light Fantastic.
Do you think it was a while ago?
It was a while ago. Sure it was. It was one of the basic sessions. It's probably...
It was one of the last ones, no?
No, for Trichuris?
We have trematodes, schistosome.
I think we did nematodes early. Like with ascaris and hookworm, strongyloides.
Wucheria, Onchocerca, strongyloides.
By the way, big meeting yesterday in...
Here we go. TWiP 20.
20! Maybe that wasn't that long ago then.
This was recorded December 15th, 2010. That was a long time ago.
It's a while back.
It was over a year ago.
Yeah.
So we are returning to Trichuris. Let's just review it.
Okay.
Whipworm, if I remember, right?
Sure.
I read in this paper that 1 billion people are infected with this.
This is correct.
Worm.
This is a member of the unholy trinity.
That's a lot of people.
That's a lot of people.
I didn't remember that. Is that the most successful parasite?
No, of course not.
The Toxoplasma is?
That's the most successful.
But more than 1 billion?
Almost everybody.
Not me.
How do you know?
Because I'm not meowing.
Did you ever be tested?
No.
No. Toxo is very, very popular.
Did you know I have no bacteria, nothing in me? I'm sterile.
No, but if you had said that as an infant, as you came out of your mother's womb, I would have believed you.
So when you're born, you're sterile?
You're sterile, totally. And parts of you remain that way.
Well, as soon as you come out, your skin gets contaminated.
Sure, but can you name, right now, there are parts of you that are sterile. Of course they are. Otherwise you'd be sick.
My brain is sterile.
Number one. So cerebrospinal fluid is sterile.
Yes.
There are 7 sterile fluids.
Is your blood sterile?
Of course it is.
Lymph?
Yes, that's 3.
But when you brush your teeth, you get a transient bacteremia.
Yeah, but that's Because we have an immune system
So
that
it's not
cleans all
sterile
that up.
then. It's not sterile.
Only temporarily.
Okay. Is your urine sterile before it
is.
comes out?
It is. That's 4.
But your 4 fluids?
Yeah.
The inside of my eye is sterile.
That's 5.
And did we say lymph?
We did.
Are my lungs sterile? No, because we're breathing air into them, right?
Right.
My stomach is not sterile.
Nope. Keep going now.
Small intestine?
It is sterile.
So in some of us, I understand.
In most of us.
Half of us?
But that's not a fluid.
Why would some people have sterile... No, it's not a fluid. Sterile small intestines and others not.
Maybe because at the time that they looked, they had bacteria in them.
I see.
They don't colonize it
So
usually.
it waxes and wanes, you're saying.
It waxes and wanes. So you've got 5 of them. You have
We
2
need
more.
another fluid?
2 more fluids.
2
more fluids. I'll give you a hint. Can you hear me?
Oh, the fluid in your inner ear?
That's 6.
Wow.
Cochlea.
Cochlear fluid, yeah.
Cochlear fluid.
That's sterile. All right.
And there's 1 more.
All right, let me just pan through the body here. I got the eye, right?
Right?
Brain, we got CSF, we got
did.
blood, we got lymph, we
Yeah.
have urine.
Yeah. There's 1 more.
Give me a small hint, not as big as the one from hearing.
Well, I would have to give you a rather risque hint.
I said urine.
That's not a risque hint.
Semen.
Bingo.
It's not risque. It's because your mind is in the gutter.
Of course it is.
Okay, I didn't even think of that. But
That's right.
you're
So that's 7 sterile fluids in your body that you maintain sterilely.
Yeah. 7, just like the number of genome types in viruses.
Just exactly like the number
Is
of
that, is that coincidental?
I don't think so. I think this is actually connected together here.
This is actually a relevant conversation, isn't it, for this episode?
Oh, no question
Yeah.
about it.
So tell me, give me the executive 1-minute
Sure.
Trichuris summary.
Easy.
Whipworm, right?
You eat an egg from fecal contaminated environment.
You eat an egg from a fecal contaminated. So where would you
Environment.
acquire the infection again?
Usually on food particles like
All right.
leafy green vegetables.
Coming
and
from a
things.
farm.
Coming from a farm in which human feces
Human
has been
feces,
used
that's
as
important. All
fertilizer.
right.
That's correct.
So you get an egg. It's a fertilized egg?
It is. It's a fully fertilized embryonated egg. It's got a larva inside.
Got it. And you eat this?
Swallow it. It hatches in the small intestine.
Yeah, it's what it says in your book.
And the
Chapter
larva,
17.
That's correct.
Larva hatch in small intestine.
And the larva comes out,
And markers to the colon then.
then it's carried down to
These
the
are
large
larvae
intestine.
now, okay.
Yeah, it carries down to the large intestine.
And they mature into adults.
They burrow into the tissue,
Uh-huh.
and then they start to grow. And they molt and grow. They molt a couple of times in the small intestine, and then they migrate down to the large intestine, where they embed into the epithelium. And the entire narrow end of this worm is woven in and out of the columnar cells.
Yeah, I remember
with
that.
the big thick end sticking out into the lumen so that the eggs gain access to the outside.
All right.
And we know very little about this worm's biology. We don't know where it mates. We don't know the location.
It mates outside of us, right?
No, it mates in the gut tract, but where?
Must be the colon, no?
Well, if it embeds
Because they haven't
itself,
matured till they got.
I
till
know,
they
but if
reach
it
the colon.
embeds into the cells in the colon, it's stuck in one place now. How can it communicate with the other males and females to actually mate? So where this worm actually
Okay.
accomplishes copulation, we don't know.
All
But
right,
after it establishes itself in the large intestine, about 60 days afterwards, and it achieves maturity, it starts to produce eggs. And the eggs, it produces about 3,000 to 5,000 eggs every day, and it can do this for 5 years.
per adult. Usually you just have
Female,
one
per
adult
female.
in you, right?
Hardly.
Several?
Hardly. Keep going up.
Thousands?
If you get that many, then you're in big trouble.
Wow, you make a lot of eggs, don't you?
Yeah, you
so
do.
you're passing these out, and then they contaminate.
They do. And they
give
can
it to
survive
someone
in the
else.
environment in a nice, warm, moist climate. They can
Right.
survive for months and years sometimes.
All right, so the Trichuris trichiura is the human whipworm.
That's correct.
And then according to your chapter, there are
Many others.
ones in the animals, but they don't infect us.
That's right. Well, I wouldn't say that.
I don't say that.
I wouldn't say that.
There's T. muris, which is mouse. You can guess from the name.
Which does not infect humans.
T. suis.
Which does
Oh,
infect
it's pig.
humans.
Interesting. In this
We'll
paper,
come back to that because it's used as a cure for something.
okay, you can remember.
I think I can remember that.
In this paper, they used T. muris,
They did. That's an animal model for human.
so they study it in
Trichuris.
mice. So,
In fact, the last author, Richard Grensis, is a good friend of mine.
is this a good model for human?
Well, as we will see, maybe
Infection?
not.
Are mice people?
I hope not.
Are ferrets people?
No. Are corporations people?
What does that mean, a corporation? No, they're not. No,
The Tea Party wouldn't let us believe that they are, though.
no. People are people.
Corporations. Only Soylent Green is people.
You know, WHO is not the primary scientific literature either.
I do know that.
Now, in this paper, they want to look at the interaction of the gut. You're hitting your microphone with the wire. You're going to
sorry.
have to stop that, Dickson.
I'm sorry.
The gut microflora with these parasites.
They want to know if there's an association. That's true.
Here's what they say. The mammalian gut contains 10 to the 13th bacteria,
Oi.
mostly bacteroidetes or firmicutes, which are phyla.
Yes,
You know those names?
I do.
And it is really so cool. Coevolution with these microbes has driven the functional morphology and immune function of the gut tract.
That is correct.
So not just how it forms, but the immune responses.
This is
It's
exactly
just great.
right. Yeah. It's why we have P450s in our liver, because a lot of bacterial products make it to the liver and they're toxic.
You got to get rid of them.
You have to detoxify somehow.
So that's the price we pay to have these wonderful symbionts in there.
If we didn't have a microbiome, you wouldn't enjoy the act of defecation.
Who said I enjoy it?
Oh, come on.
You-
Read
you're
Ulysses,
just
for
awful.
gosh sakes.
Stop. Listen,
That's
it
my
says
pick
here,
of the week, by the way.
we don't have picks on TWiP.
I want one.
It says here a Helminth-driven Th2 and regulatory helper T-cell response has evolved to counter infection and repair the damage that these parasites cause. So it's driven by helminths.
Yes.
They also say helminths have been found to be a major force underlying the evolution and selection of interleukin genes. Did you know that?
I did know that. Of course I did. I'm a nematologist.
I think we have discussed that before,
We
or
have.
thought about
We
discussing
have.
it.
Peyer's patches, all those things that you see along the gut tract to counteract the presence of microbes were primarily engaged in anti-helminth activity.
I see.
And eosinophils and IgE.
Mm-hmm.
All of those arms of the immune system come to bear on these large critters.
Okay. So the way they justify this work is in the following context, Dickson.
Yes, Vincent.
They say, so gut commensal bacteria and helminths have lived in close association, and the relationships between bacteria and metazoa have been documented, like worms and Wolbachia. Remember we talked about
Exactly
that?
right.
However, a functional relationship between prokaryotes and parasitic metazoa within the host remains to be defined. So Trichuris is a parasitic metazoan,
Of course.
correct?
Yep.
Multicellular
That's exactly
worm.
right. Yep, sure is.
So that's what they've done here.
Yep.
A few more factoids. Besides infecting 1 billion people,
Yes.
it says it comprises more than 50 species, this Trichuris genus. Is that right?
Yeah.
50 species?
At least.
Billion people. And listen to this. All Trichuris species inhabit the large intestine, cecum and colon.
Correct.
Inhabit. But as we said earlier, the larvae hatch in the small intestine.
That's right.
So by inhabit, this means the permanent
Yeah. Where the
resting
adults
place.
live. Where the adults
All
live.
coiled up there in the
That's right.
mucosa.
That's
Okay,
right.
so
That's
that's correct.
Yep.
So he says here we investigated a bacterial-driven mechanism of hatching of the mouse whipworm that
Cool
also
stuff.
influences the subsequent immune response.
So there's a good friend of mine. He used to teach
Hatch.
at University of Nottingham, which is where Richard Grensis got his degree.
Yeah, this is from the UK. Did you know
Yeah, he
that?
got his-
Manchester.
That's right.
Yeah.
I actually saw him last summer when I went over to visit at the
Really?
Manchester International Festival.
Did you talk about this work?
I did. In fact, we had a nice discussion.
Should have
about
brought a
this.
recorder with you. You could have recorded a podcast.
No, we should have got him on here Skyping.
Yeah, it's true. But he's
He's a little bit away from
it's
us.
evening now.
That's right. Did you know that there's a park in New York City that celebrates Trichuris infection?
Is that the one with the worm in the grill work?
Yeah, we're going to put that up.
You
as
showed
a-
me that last
I-
year.
I
Remember, you
did.
took a picture of it.
Let's put it up as part of this
I
podcast.
think I put it on Facebook.com/thisweekinvirology, but we could put it here
It's
as
very
well.
appropriate to go here.
That's a Trichuris?
That's Trichuris.
Wow.
The male. It looks just like this.
Yeah. That's interesting.
Yeah.
So what they do here, Dickson, they're talking about hatching
I know.
these eggs. Which
they're also
for
talking,
me is for chicken eggs. You
they're
know, that
talking
shell.
about site selection also.
Site?
Site selection. Where does this worm live?
All right, so here's the shtick, Dickson.
Yeah,
they
yeah.
take explants of mouse cecum. Now, you've worked with mouse ceca before, right?
I have, except
So
the
what
Trichinella
is an
doesn't.
explant of a mouse? You cut it out and you make an extract, which have a lot of bacteria in them,
Or
right?
you leave it intact
And they say they put the eggs. Where would you get T. muris eggs from?
from a T. muris infection, Vince.
You have to purify it from the feces?
You do. It's easy.
It's easy?
Yeah, it's very simple.
How?
You use a high-density sucrose solution.
Okay.
And the eggs will float under certain conditions, specific gravities, and the rest of the stuff will go to the bottom. So you can purify the
Nice.
eggs out of it very easily.
So they say these cecal explants have a lot of bacteria in them. And if they mix
Yeah, they
this
do.
with eggs, the eggs hatch. 37 degrees, 30 minutes, the eggs.
In the extracts.
How do you tell if a T. muris egg is hatching? You just look at it?
Yeah, under the microscope, it has two little pores or capsules or little covers. They're
At the
called
ends?
opercula.
Yeah, that's right. Look at
Looks like a
There they
football.
are.
It
I
looks
think
like a football.
we talked about
We have.
this
They're
before.
two little manhole covers. Just think of them as manhole covers.
Person hole covers,
Right.
Dickson.
Sorry, I have to degenderize
Yes.
this. You're absolutely right. So, in either direction, depending on which way the head is pointing of the larva inside the egg,
Right.
all right, there are hatching enzymes that are elicited by the environment, which cause the enzyme to become active, and it actually etches away the covering holding, the glue
Oh,
that
okay.
holds it in place. And
Nice.
so out comes the worm.
So here, these you mix them with this cecal extract.
Yep,
They hatch. So then
they do.
they said, what bacteria? So they tried E. coli.
Right?
It works.
Okay.
Mix them with
With a pure culture of E. coli,
yeah, it hatches.
it hatches.
And if they filter the E. coli culture
Right.
and use the broth, it doesn't work.
No.
Which means why?
Probably some physical association.
And not just a soluble factor
Not just a
or a virus.
or a virus that kind of make you sick.
Then they tried different strains of bacteria. They say 5 different strains of bacteria, and one yeast will do the same thing.
Interesting.
So it's pretty diverse.
Interesting.
You have yeast in your gut, Dickson?
Shouldn't have them, but you do sometimes.
Sometimes?
Yeah, it depends on what you've been eating
They also
or drinking.
show that you need direct contact between the bacteria and the eggs. You know how they do that? I bet you know this.
Tell me. You'll tell me anyway.
They use a transwell where you can put the eggs on one side and the bacteria on the other of a thin membrane, and that's no good.
That's
If it
an
were a
engine.
diffusible factor, it could go through this,
That's true.
but they have to touch.
We used to call those rose chambers.
Rose, R-O-S-E,
Yeah, you know about
named
them?
after Charlie Rose,
That's right. No, not after Charlie
the
Rose.
talk show host.
Sorry. No, rose chambers.
Were you on the Charlie Rose show?
Never. No.
Let's see, what else? This is very cool. They have an E. coli that
Yes.
produces GFP.
What?
So they can do these microscopies and see. This way they have a few of these pictures here. And you can see
Sure.
the E. coli are green in the picture.
Yeah.
Where are they binding?
Well, I can look around the corner here.
I'm sorry, Dixon.
That's
There.
okay. Are they
Look.
binding? They're binding to the eggs.
Yeah, where on the eggs?
Oh.
Look. Can you
They're
see them?
binding at the poles.
Yeah, those two opercula.
The two opercula. Oh,
Both. They cluster around the opercula, they say right here.
They cluster around.
This is
the
real
opercula.
life parasitism.
So that's a receptor
Maybe.
and a molecule that's a
Yeah,
ligand.
maybe. I mean, I don't know how these. I don't know what these eggs are like around them. If there's something specific for the opercula, or it must be, right?
Yeah, but it's from a bacteria, though, Vince. Does that make sense to you?
What's from a bacteria?
The attachment point of this egg to its host is mediated through a bacteria that acts like a link.
Why do you say that?
'Cause that's what they're going to tell you.
Okay, we'll see. Next, they boil the bacteria.
Right.
What doesn't work. They don't hatch the Trichuris eggs.
No.
So that means they have to be living.
They've destroyed something. They've denatured something by boiling it.
And they ask if it's the fimbriae of E. coli that are doing it. Why do you say that?
Could it be?
Fimbriae? What is a fimbriae?
Filamentous projections from the surface of E. coli.
So they they treat them with
Those are not pillae, though, are they?
no, they're not pili.
That's right.
Type 1 fimbriae.
These are different.
They add mannose, which would inhibit, which would bind to them.
And
That inhibits
binding to the operculum.
hatching.
Right.
And then they have a strain of E. coli with a mutation in the gene cluster that produces Fimbriae. It doesn't make Fimbriae, and these guys don't.
So they've
they don't
narrowed
hatch.
down the linker between...
Well, but listen to this.
I'll listen. Shut up and
The
listen.
gram-negative bacterium Pseudomonas aeruginosa does induce hatching, but it doesn't express type 1 Fimbriae.
Oh.
Similarly, Staph aureus promotes hatching, and there's no Fimbriae there either. So,
And yeast don't have Fimbriae, and
No.
it says it doesn't
yeah,
also.
that's right.
So there must be something else.
Something
that
else.
would work. Maybe
Then-
it's the mannose that matters.
Salmonella exhibited mannose-sensitive hatching.
Ah.
So they have Fimbriae. But,
because they're related to E. coli, because they're
But
Enterobacteriaceae.
so Pseudomonas induced hatching in the presence of mannose. They don't have Fimbriae, so the mannose hits the Fimbriae.
But it must have hit something else on this bacterium, which acted like a bridge as well then.
What hit something else?
Mannose. The mannose must have linked the bacteria to this operculum through another molecule.
It inhibits hatching, though. The mannose.
Oh, okay.
Then they take this Fimbriae protein. It's a single protein. It's called FimH. They put a histidine link on it, and then they mix it with the solubilized eggs, and to see what proteins the Fimbriae are binding to.
Solubilized eggs?
Yeah, they call it solubilized surface egg proteins.
Oh, okay. All right.
And then it says 5 to 7 surface proteins were detected that bound to purified FimH.
Huh.
And they're in the process of identifying these. Of course, this paper is June 2010, so they probably had done it by now,
Yeah.
but I didn't look.
No.
Why would you want to know what proteins are being bound? Where's this going to lead?
Well, you might be able to intervene with that somehow, like with a lozenge. This is an
But
aside.
we have drugs to treat this. We don't need anything else, do we?
Yeah, but then you have to get sick first.
Why
This
is
could
that?
prevent it. This could prevent
A
infection.
preventative?
It could prevent infection.
Hmm.
So there's this company that was developed as the result of discovering that bacteria use various glycosyl residues to attach to the gut tract.
Yeah, right.
And the guy who formed this company actually worked out a number of attachment mechanisms that pathogenic microbes use to attach to our gut tract,
Okay.
and developed a preventative lozenge
Lozenge, yeah.
that you could suck on all the time that you're at a meeting, let's say. Let's say you go to a place where you know you're going to get sick because you have to drink the water and the water is contaminated. You suck on this lozenge, and it releases all these sugars that
I
competes
see,
out
yeah.
the bacteria. And
It's like taking chloroquine when you're going to a malaria endemic region.
yeah, but that's going to kill the microbe. In this case, it's not going to even allow the microbe to attach, because that's the first step in the infectious process, right? Even with viruses, it's the same thing, right?
Yeah.
So you need a lozenge for viruses as well as for bacteria. Now it looks like you need a lozenge for parasites,
If it gets that far, I don't know.
parasitic eggs. Some of the eggs don't work like that.
Then they say, we don't know if this has all been done in vitro, right? So what do you need to do next?
You got to move it into the animal.
Into a ferret?
No, into a mouse.
I gotta get away from this ferret business. It's driving me crazy.
I think that's true. You got to weasel your way out of that.
I'm obsessed with these ferrets. So they looked in mice. They treat the mice with antibiotics to alter the gut flora.
Right.
And what they so then I guess what you do is you treat the mice with antibiotics, and then you feed them
Trichuris eggs.
eggs, and then you see
Right.
what do you do? They say the worm burden is
Right.
reduced in animals treated with antibiotics.
Yeah, the number of worms that establish.
How do you
after
count that?
Well, you have to wait a while because it takes a while for these things to grow up.
Yeah. So then you have to cut the mice open. And,
Yeah, and then you have to examine them and see how many worms are there.
Okay.
But these are big enough so you can see each worm, right? They're all visible. And this worm does live in the cecum, by the way.
So these mice, they say, make a stronger Th2 response. So T helper type 2 cells that produce certain kinds of cytokines, they have a stronger response. So apparently the increased worm burden in a normal mouse, not treated with antibiotics, suppresses the Th2 response.
Isn't that
interesting? It's manipulating
Huh.
the Th2 response.
How dare
They
it?
also say that parasite-specific antibody, IgG, in the blood was increased in antibiotic-treated animals.
Huh.
So you reduce the parasite burden, and you actually get more antibodies against the parasite. So the parasite is suppressing responses against itself to allow its persistence, right? Does that make sense?
It does make great sense. That's why they're so successful.
And then they also look in immunodeficient mice, SCID mice, which lack B and T cells.
Right.
Because they want to make sure these effects on the immune response aren't interfering. So they take these mice and they treat them with antibiotics as well, and they also see worm burdens are reduced in antibiotic-treated mice. So it's the actual bacteria, not the immune response, that's altered.
So, Vince, why do SCID mice survive at all with bacteria?
Well, clearly they have bacteria.
I know they do. Why don't they die from that?
I don't know. Why don't they die?
Because they have innate immunity.
Is that enough to protect them?
It must be. They have NK cells.
I don't know. You know, that's a good question. Some of our listeners may know the answer. Is
Right.
innate immunity enough? Because these are lab mice that are growing, and
that's right.
they're given normal feed. It's not sterile.
Sure.
And they obviously have gut flora. You know.
Yeah. So nude mice also have been used a lot in parasite experiments as well. And I don't know what would happen with those. You can't keep those alive very long.
Well, you have to put them in a barrier facility, right?
Yeah, and they're best raised under germ-free conditions.
Okay.
But that would be the ultimate. You take a germ-free animal that's normal in every other way except no bacteria at all, and then will this infection survive? Will it actually take? So they didn't do that experiment.
No, they didn't. There's plenty more to do.
I did that experiment.
Yeah,
I did.
With Trichinella.
Yeah.
Hey, did you ever see if I think we've talked about this.
We
So
have.
in germ-free mice, you give them Trichinella.
Yeah.
You
get a
abandon
weird
that.
result. You get a weird result.
Okay, so then you started looking at other things as a
did.
result.
I
But
mean-
maybe you were onto something and you should have pursued
It
it.
may be. Luck favors the prepared one, and I was certainly
You
not
were not
prepared. I wasn't prepared to get lucky on that day, that's for sure.
So their conclusion is the paradigm that the adaptive immune response system has evolved to control microbes
Right.
has been modified to reflect the role of microorganisms in its development. And they say we have to extend this to include macrofauna, the metazoan parasites such as Trichuris. Although not everyone has this in their gut tracts, Dickson.
That's
so,
today, but in the old days.
yes.
You see, they're using this approach to account for the fact that today, since we don't have these macrobes rather than microbes, we still have the microbes, but we don't have any macrobes. We now have a number of replacement disorders, and most of them relate to allergies. And Crohn's disease is one of those big ones, the autoimmune disease. It's partially curable by reinstating the macrobe aspect to their gut tract by giving them these Trichuris infections. So the target has been removed by sanitation. And so if we replace that target with portions of the worm, I think we'd have a great chance at permanently curing Crohn's disease. Because you can cause a remission in Crohn's disease by simply giving them Trichuris ovis, which is in the sheep, or Trichuris suis, which is in the pig. You can actually reduce or eliminate Crohn's disease during the time that the worms are present. But of course, your immune system will eventually kick them out. And when that happens, Crohn's disease reappears. And in fact, if you go to Africa, into these villages where 90% of the kids, in this case, have Trichuris, ascaris, and hookworm, those three together, that's why they're called the unholy trinity. And you look around for Crohn's disease, you can't find it. It isn't there. If you look for food allergies, it's not there. If you look for rashes and skin allergies and pollen allergies and grass and ragweed allergies, it's not there. They don't have any of these problems. They've got worms, of course. And the moment you remove those worms and take those kids and move them to a clean environment and keep them that way, they start to develop allergies. And that's very interesting. Because the reason why I'm mentioning this is that the Gates Foundation and the World Health Organization yesterday announced a multi-pronged $785 million prevention program or elimination program for the top 10 tropical infectious diseases, among which were lymphatic filariasis, schistosomiasis, hookworm disease, ascaris, and Trichuris. Those diseases can be eliminated using multi-drug approaches and sanitary approaches.
Yeah,
And the
yeah.
prediction is that once those disappear from areas where they're now endemic, these other autoimmune diseases will start to appear, and you'll have other medical problems to deal with. So, I mean, it's it's you're damned if you do, and you're damned if you don't.
So they're arguing that this presence of these
Yes,
Trichuris are important for normal balance,
exactly.
right? But if you have too many, you get sick.
You do.
So what do you do?
ordinarily, if you take a spectrum, I meant to tell. I don't know if we've discussed this part or not, but I'm about to draw something on the board, but we can get some diagrams for the show as well. So, Vince, imagine that I'm going to block out now the population of a village. Let's say in Central Africa. Let's say in Uganda.
I should videotape this.
Nah, you don't have to do that. But I need to erase something then. You don't mind? You mind if I erase this?
No, I don't mind.
Okay, I'm going to erase this little.
I could put this little movie in with the show notes,
Okay, let's really.
Because I do have a video camera right here.
Oh,
in
this
my
will be
pocket.
good. This
Hang
will
on
be good.
before you start.
All right. So I'm back in the classroom.
All right, smile.
I'm smiling.
All right, now do your thing on the board.
Okay, so Vince, let's do this. These are the number of parasites.
Does it make you feel like the old days?
It does. And these are the number of people, okay?
All right.
So, and these are the number of parasites. This is one parasite, 2, 3, 5, 10, 20, 40, 100, and, well, whatever. Let's make this a log scale. So this is 1, 10, 100, 1,000.
Let's see how far Dickson can go.
I don't have to go farther than that. That's 10,000 is next. And these are the number of people in the village with one parasite. Okay, the number of people in a village with one parasite, let's say, these are percentage now. This is a percentage. Let's not make it a number. Let's make it a percentage. And let's say this is 50%.
With one.
50% of this village have one parasite.
So they're happy because they have a good immune
Or
response.
1 to 9 parasites.
All right.
Another percentage of the village has more parasites, and another percentage has more. And a very small percentage over here has most of the parasites. So this says 90% of the parasites, number of parasites, are present in less than 1% of the people.
And those are the problematic people.
These
They're
are the people.
Yeah, they're sick, right?
Yeah, there's a horrible term for them, which I hesitate to use, but they're called wormy people. So if you could identify these people,
Yes.
there's a genetics to this, by the way.
Yeah, of course.
Not just with the parasite, but with the people.
People, yeah,
The
sure.
susceptibility to these parasites is unevenly distributed throughout the population. So as well as the fact that these parasites modify your immune system,
Yeah.
there are other people that have an unmodifiable immune system.
Right.
And those are the ones that we wish we could identify before they got infected. Because these people are giving these parasites to everybody else.
You would think that those individuals would be selected against, right?
You would think so, but you know what? There might be some advantages to having all those parasites. There might be.
All right.
And it's true, by the way, this same curve is obtainable for Ascaris, for Trichuris, for hookworm, for schistosomiasis, and for malaria. And each one of these groups is unique. It's a different mechanism in each case, but it's the same curve. That's remarkable. That's truly remarkable. So every parasite that's reacted against by your immune system is very complex in overall complexity, and the protective mechanism is unique for each species.
All right, so in the U.S., we don't have this infection at all?
Wouldn't say not
Very
at all.
little.
Remember, the egg has to sit in the soil for a week before it's infectious.
Yeah.
So we have good sanitation for human feces, human fecal contamination, I should say. We don't use it as fertilizer. We don't randomly defecate wherever we go. You'd have to go to the Caribbean to find out for endemic centers.
So the question is, what are we doing to our immune systems by having removed these parasites?
That is the major, big, giant question.
the hygiene hypothesis.
That is.
We're messing things up, right?
Well, I wouldn't say messing them up. We're changing them.
Well, the increase in asthma may correlate with decreases in...
Do you have any allergies, Vince?
No, I don't have any.
I do. Ask me what I'm allergic to.
What are you allergic to?
Mice.
Mm-hmm.
I got that allergy by working with them and then staying away from them for a while and then coming back.
I don't have allergies. Maybe I have a good gut flora. Maybe I have Helicobacter, but I'll have an ulcer soon. I'm at the age.
Maybe.
What should I do?
I don't know. Go for regular checkups, exercise more, lose weight. I don't know. I can't tell you what to do. Eat a balanced diet.
Well, I feel fine now. I don't have any pain in my stomach, but the thing is, once the pain starts, it's too late.
Don't worry. The point is that don't worry. Just don't worry, all right? Because we can spend all
I
our...
worry about a lot of things, Dickson. Anyway, let me tell you one more thing at the end of this paper.
Please.
You said something about connecting the parasite and the bacteria to the host.
Yeah.
I didn't do any experiments to address that.
No, they didn't.
No. But here's what they say at the end.
Yeah.
We clearly see a critical relationship between bacteria and T. muris that has a marked effect on the host immune response to the parasite. We propose that temperature, together with an increase in bacterial load at these sites, provides optimum conditions for efficient hatching of Trichuris species eggs. Additionally, controlled damage of the intestinal epithelium provides an enhanced potential route of access for microflora to the adaptive immune system. This has subsequent beneficial consequences for the host in that antibacterial Th17 responses and Th1 responses are generated, various T helper responses that are known to control pathogenic bacterial pathogens. That's a funny sentence, pathogenic bacterial pathogens.
Exactly.
Dickson.
Yes, Vince.
So there's a role for Trichuris in good immune responses, and then they're also saying there's controlled damage of the epithelium. By whom? By Trichuris? So it's helping bacteria, helping us to make an immune response against bacteria that will protect us.
It's damaging a little bit to allow entry points for the bacteria, which then stimulates the immune system.
That's interesting. So if you give this to people with Crohn's, what is the effect? It is suppressing the immune response, which is part of the pathology?
It doesn't suppress the immune response. It redirects
Redirects it. Okay.
Instead of reacting against a tissue antigen, you're now redirecting it to the worm itself.
Now,
let me ask you this, though, Vince,
Yeah.
because you've asked me a lot of questions. I want to ask you one
I always
now.
ask questions.
I know you do. So I have a question, and it's a generic question, and we have to think our way through this for a while. How can a worm live in the gut tract for 5 years like that without being eliminated? And then finally, of course, it is. But how does it live that long and survive the attack that obviously is going on all the time?
I don't know.
Neither do they. They're very interested in answering that question.
Mm-hmm.
They really are.
I have a question.
Please.
These eggs hatch in the small intestine.
No, they hatch in the cecum in the mouse.
Oh, it's different in the mouse?
Yeah.
How do you know this?
Because they told you.
So in humans, the Trichuris hatch in the small intestine, but in the mouse, they hatch in the cecum.
That is
just
correct.
Another reason why mice are not people.
That's right.
Okay, thank you.
You're welcome.
That clears that up.
Yep.
Any other tidbits before we move on?
Well, just to mention the fact that the small intestine in most people is usually devoid of bacteria.
Yeah, I was looking at a study before, and they had found in half of the individuals it was sterile, and half there was some colonization. I understand that colonization of the small intestine is associated with disease.
It is. Otherwise, it shouldn't be inhabited by bacteria.
All right. You want to read some email?
Sure.
This one is from Trudy. I almost caught up listening to TWiP. I look forward to your future efforts, ebooks, etc. However, in TWiP 32, I thought I heard you mention that there would be a link to Dr. Despommier's lectures. I couldn't find them, so I sent them to Trudy.
Good.
I am dreading listening to number 33 because there are no more TWiPs available yet.
Oh.
I live in a semi-tropical climate in southwest Florida, so tropical diseases and parasites are especially interesting. The only good thing about being current with your series is that I can start to catch up on TWIVs and TWIMs. I did find Dr. Racaniello's lectures in virology. I started with Delta virus.
All right.
Dr. Despommier, please consider doing a podcast on vertical farming and even throw in some other tidbits of gardening wisdom. I took the user survey. Perhaps the results will help you in your future efforts. The way the you two work together on TWiP makes for a delightful experience. Science, medicine, and pathogens float my boat, so it is a great pleasure to hear two learned professors so engaged in interesting discussions. Then she says, P.S. She's a former public health nurse from Naples, Florida.
Oh, yeah.
Since you have both a sense of humor, if you should happen to read my letter on your podcast, My last name is pronounced like what you would not want to have in any kind of car, let alone a Bentley. I sometimes tell people, what?
You're not going to read our last name.
that I used to be a Bentley, but when I married my husband, he made a wreck of me. Thank you for I sent her the link to your lectures. Thank you for the reply. I watched the first lecture last night. You both make me want to go back to college. I'm 64 years old, so my studies are self-directed via venues like TWiP, TWiV, and TWiM. It is my vow to continue to learn until the very end of my life. Please share my gratitude with Dr. Despommier.
Marvelous.
Peter sent us a link to an article. Look at this, Dickson.
I will look.
10 most diabolical and disgusting parasites.
Yeah, no, I've seen that, actually.
You have?
Yeah, I have.
What is that? It's not a parasite, is it? It's a huge bug in the fish. We've talked about this before, haven't we?
A parasitic louse.
Really? We took a look at the next one. Sexually transmitted cancer in dogs.
Right. Oh, come on.
First dinosaur was killed by Trichomonas. Sue.
So they say.
Yeah. And then one more. Oh, here's the parasitoid wasps.
Oh, yeah. Okay.
So we'll put a link to that.
The ichneumid wasps.
Would you like to read the one from Brian here?
I would if I could.
Can you see it?
Stretch around this.
That's the best I can do, because if you move it anymore,
That's
you're
right.
going to disrupt the recording, Dickson.
Okay. It says, Vincent and Dickson. My wife pointed me to TWiP, and what a blast. My 23-year-old son and I are making our way through the early episodes during our commute to and from work and loving every minute of it. After listening to the tapeworm episodes, I will never pet a dog from Wyoming or any other of the sheep states. I am not sure if you have covered the evolution of parasites. We're only up to episode 9, but that must be as crazy as ever. Surely you have enough material for several years' worth of podcasts, but I'd like to hear a bit about how an animal accumulates such a bizarre life history. Cheers. Well, that's a great idea. We could discuss not the origins of parasitism, because we've had a few discussions about that in TWiV, but we could discuss how parasites and hosts co-evolve, because there is a lot of data on that out there now.
Are there experts that you might know who could join us?
There are, by the way.
Okay, some.
We'll have to get in touch with them.
Thank you, Brian. Next one's from Peter. Dear TWiP team, I was speculating about parasites that modify host behavior and a possible strategy that may have evolved in those transmitted by blood-feeding vectors. Mm-hmm. To achieve optimum transmission of the parasite, uninfected blood-feeding insect vectors should find infected hosts more attractive than uninfected ones, and once the insect vector had itself become infected, its behavior should change so that it would then preferentially seek uninfected hosts. Is that correct?
It's one possibility, but there are many others, and including the fact that a sick person moves a lot slower. And let's say you're suffering from malaria and you're in and out of consciousness, you won't even be awake enough to swat those mosquitoes that are feeding all over you. So that's probably a more likely scenario as to why infected hosts are more likely to be bitten, because they are not. They are at rest. They are not moving, and they're not conscious of their environment.
He asks, has any research been done to determine whether any parasites that employ blood-feeding vectors that actually do modify host smell and vector preference in this manner?
I don't know of any.
No.
Sorry.
As I was writing, I was thinking of arthropod vectors such as mosquitoes and biting flies, but it occurred to me that there are other possible blood-feeding vectors such as vampire bats and leeches, whose behavior could also be potentially modified in this way. Are there any zoonotic infections that are transmitted by leech vectors?
There are, but not to humans. They transmit a trypanosome infection among lizards. And bats, of course, they transmit rabies, but I don't know of any other parasites that are transmitted through the bite of a bat. Well, not the bite of a bat, through the droppings of a bat. We know about Nephovirus.
Lastly, I have seen blood feeders described as hematophagous and sanguivorous. Are these synonymous? And if not, what is the correct term?
No. Hematophagous is the word that most entomologists apply.
But not sanguivorous. It means the same thing?
It means the same thing.
All right. Peter is from Turkey.
Nice.
Gopal sends us a letter from...
No, go ahead. Yeah.
From Science. It's just an interesting article.
Yeah.
He says, can't help wondering somewhat uneasily what our own parasites make us do. This is entitled Animal Mind Control.
Oh, yeah.
Examples of parasites that manipulate the behavior of their hosts.
Right.
Yes, this is common stuff, right, Dickson?
Well, not so common, but when it occurs, it certainly draws our attention.
Pet cockroaches.
Of course, toxoplasma.
Fatal attraction, the
That's right.
toxo business.
Toxism,
Yeah.
big deal, yeah.
Swim towards the light.
Yep.
This is compelling for people, isn't it?
I think so. I think this is how you learn the interlocked nature of the physical and the biological environment. We are driven by natural cycles.
Yes.
And I actually watched a fascinating show on the Discovery Channel once about what would happen to life on Earth if there was no moon.
How about no oceans?
No, there is a show called Train the Oceans, but that's just to show you what the bottom of
What
the
would
oceans
happen
look
if there were no moon?
Very, very horrible things would happen, Vince. First, no tides. Right?
Okay. No nocturnal animals.
No nocturnal animals. Well, certainly not that are diurnal in terms of their habits of migrating
Curious.
to and from. A lot of other things would happen, and in fact, the moon is a shield. Notice how pockmarked it is with meteors that have hit it. Those would have hit the Earth probably if they hadn't hit the moon.
Yeah.
So that it's sort of like a natural shield that works around the Earth.
Interesting.
A lot of other things they predicted. And by the way, it seems like a fanciful thing to talk about. What would happen if there's no moon? The moon is in retreat, meaning it formed a lot closer to the Earth than it is now. It's 240,000 miles away. When it formed, it was only about 80,000 miles away.
Wow.
It must have been frightening to see a moonrise.
Huge.
It was
cool.
It took up almost half of the sky when it rose.
Neat.
Yeah, neat is right, but imagine what the tides were like
Didn't
then.
obscure the sun.
It could have. Of course it could have. The other thing, of course, it's now in retreat.
How far is it going to get?
It will disappear. It's going to get out of Earth's gravity soon.
How long will that take?
They said about 2 or 3 more billion years. And
All right, let's move on.
a short
billion
2 or 3
years.
billion years.
Read the one from Don.
All right, Don. Don writes, in your discussion of Wolbachia and filariasis,
So you need to speak with force.
Vincent suggested tetracycline for therapy. Dickson opined that it was too toxic and expensive. Au contraire. In the QID form, it is dirt cheap, and it has been used widely since the mid-1950s. Prolonged use does cause mottling of the teeth and fluorescence of the bones under Wood's light, but not a problem for most of us. Wonderful podcast and deeply appreciated. Thanks. Okay, I sit corrected.
Yeah, you're sitting.
And I'm corrected. I always take useful, correct criticism to heart.
Okay. Kyle writes, top of the morning to you.
Must be from Ireland.
No, he said, I'm not Irish. I just like the phrase. I want to thank you for putting together such an excellent podcast. I'm currently a med tech working in a parasitology laboratory.
Oh, great.
And enjoy my days searching through blood and poo.
My first
for
job.
Those tiny life forms that you describe so well. You must
I do. I
identify
deeply
with them.
associate it with them.
And vice versa.
Oh, good.
I was introduced to the world of parasites by a course I took as an undergrad. The course instructor was Dr. James B. Jensen.
Oh, a good friend of mine.
Is that right?
Yeah.
Judging from your earlier talks, I trust that this name will sound familiar.
It's more familiar than you think, because as I'm turning the pages of my book.
And as you can imagine, the course taught by such a well-known parasitologist was an amazing experience. Oh, he wrote the introduction to your book, Dickson.
He wrote the
forward.
He wrote the forward.
I have a great passion for parasitology, and I'll see what kind of future I can put together. Thanks again for the great shows. I encourage you to continue and add my voice to those that are calling for more. It is this week in parasitism, you know. Yeah, I forgot.
Our clock is ticking a lot slower than yours. If you do see Dr. Jensen on your travels, say hello to him for me. I used to trout fish with him, in fact.
The next one is from Jim, who says, delighted to hear the latest TWiP on the malaria vaccine. You guys mentioned using impregnated bed nets to reduce malaria.
Yeah.
The New York Times had an article describing using poisoned bait to reduce the mosquito populations by a bunch like 90%. Would love to hear your thoughts on this. What do you think, Dickson? Donald McNeil, one of my favorite science writers.
Oh, nice.
Not really. He said that the fatality rate of H5N1 is 60%.
That's not. It's the case fatality.
And I wrote him, and he said, I don't believe that data.
You don't believe that there are inapparent infections?
No.
That's ridiculous.
So have you read this?
No.
What's your opinion?
I have not read it, so I can't give you an opinion. I think they've used poison bait traps for other insects that spread diseases like tsetse flies, and it works. But tsetse flies are less abundant in nature in many areas. In fact, in East Africa, one tsetse fly per square mile.
Mm-hmm.
That's how not dense they are. So if you kill off one tsetse fly, you've cleared a square mile's worth of savanna of sleeping sickness. So
they're using poison nectar here.
Poison nectar, I mean, it's a great idea, but it would be, because that's what the mosquitoes feed on, not blood, but rather plant nectars. So that's the correct food to give them. I would worry about other things feeding on them, like hummingbirds and other insects that feed on it as well. I would worry about an ecological ripple effect. That would be my big worry.
90% die-off in a study in Mali. Isn't that where your friend at the NIH has an outpost?
Dr. Robert Gwatz, who was on our show, of course.
I wonder if he's involved.
I don't know.
It kills insects that eat it.
Yeah, but not just mosquitoes, though, right?
Insects, cockroaches die.
Understood, but how about normal insects?
Doesn't say anything about that.
What about the pollinators for the crops over there?
Yeah,
something to worry about.
They got $1 million from the Gates to do this.
I understand.
Wow.
And did they say which group did that?
Yeah, Karen Altman.
Oh, yeah.
And this fellow at the University of Wisconsin Veterinary Medicine School. Oh, he wasn't involved.
Bruce
he said it's a cool
Christensen.
thing.
I know Bruce.
He said it's a cool thing.
I know Bruce.
But where are the others? Where are they located?
All right.
The University, Hebrew University in Jerusalem.
All right.
Do they have malaria in Israel?
No.
Why do you think they would do this?
Because they're concerned.
Okay.
This is a good idea. I mean, I don't think it's not a good idea. I think it's a good idea as long as it doesn't have this ecological ripple effect. That's all.
We was also delighted to hear that you guys are going to have more TWiP.
We are doing that as we speak. In fact, as we speak, we are TWiPing.
Still devoted to TWiP MV. Thanks much in 73.
Ha! Very nice.
Want to read Kelly's? Are you able to read such a long letter, or is that too much for you?
I can read long letters. It's just from this angle. It's a little bit challenging.
Why are you complaining?
I'm not complaining.
You want everything perfect in life?
Hi, Vincent and Dickson. I'm a PhD student at the University of California, Davis, and a longtime listener of This Week in Parasitism and This Week in Virology. I have a great appreciation for the fact that you two are successful scientists who take time out of your busy schedules to share your love of parasites and viruses with the general public. In fact, I was so inspired by This Week in Parasitism that I have since begun podcasting about science topics myself. Thanks for the inspiration. I thought you two might be interested in an experiment in science crowdfunding called the Sci-Fund Challenge. One exciting aspect of this experiment is that we're exploring a potential new source of funding, i.e. the general public, for small science projects at a time when everyone seems to be complaining about how difficult it is to get grant money. While this aspect of the experiment is certainly exciting, the most exciting part to me is the fact that crowdfunding forces scientists to engage with the public. Not only does the public get a chance to learn about cutting-edge research, but they get the opportunity to have a voice in deciding the direction of future research through the money they donate. While I think you might be interested in learning about a program incentivizing science engagement with the public, my motives are not entirely altruistic. I have a Sci-Fund project focusing on how trematode parasites manipulate the behavior of their hosts. Wow. And I'm wondering if you would be willing to help me get exposure to my Rocket Hub proposal for this project. URL is here. Or go to rockethub.com and search the brains of support zombie research. I study Euhaplorchus. Euhaplorchus. I'll try this again.
Never heard that one before.
I study Euhaplorchus californiensis. It's EUAH. HA rather, EUHA, a trematode parasite infecting the brains of California killifish, which is a brackish water fish that lives probably in the Sacramento area delta. EUHA manipulates the neurochemistry and behavior of their fish hosts, a fact which I find endlessly fascinating. These parasites have essentially had millions of years to figure out how the brains of a fish work, and to figure out how to manipulate brain chemistry to achieve their own goals. I'm working on quantifying the extent to which the parasites modify the behavior of their host, and am trying to figure out the mechanism by which these parasites achieve their extraordinary behavioral manipulation. I believe parasites have a lot to tell us about how brain chemistry influences behavior, and I think of my research as an exercise in coercing EUHA to tell me what it has learned over millions of years about brains and behavior. If you would be willing to tweet about my project or mention it in any other way, I would be endlessly appreciative. I am happy to answer any questions you might have about my study system, and it's pretty much my favorite thing in the world to discuss. So
Cool.
We just featured your research quest for funding as an email read. How's that?
Sounds good to me.
It does.
Let's see how we're doing here. We are at 60 minutes, a couple more. Next one is from... Oh, I just lost my place. Trudy.
Whoops.
Oh, here's Trudy again.
Okay.
I'm a registered nurse with an unusual background. I worked as a diener at a teaching hospital while working my way through college. My mom was an RN. Also, when there were few vaccines, she graduated in 1935. She would often discuss what she had seen as a public health and army nurse anesthetist in World War II.
Wow.
She had a library of medical textbooks that I enjoyed reading. I spent part of my career as a nurse epidemiologist, county public health practice, and as an STD clinician, where I got to work with all kinds of infectious diseases and conditions, viral, bacterial, and, of course, parasitic. I have been interested in infectious diseases and conditions since I was a young child when I got my first microscope. I've been lucky enough to live in semi-tropical Florida for over 30 years. The University of South Florida still has a great series of podcasts from their medical school's Division of Infectious Diseases. They also discuss tropical medicine. My first in-person experience with a parasite specimen, which really hooked me on microbiology, was that of a tapeworm, which was in a huge, tall container in the foyer of Abbott Labs in Chicago. I was with a group of high school students which had an NSF grant in collaboration with Northwestern University's College of Medicine. I've been fortunate to see a lot of interesting things because I also did some biomedical illustrations. I'm both an artist and a nurse. To make a long-winded email shorter, your podcasts are infectious. I believe there is no cure or treatment other than continuing as a listener to you discuss microbes in your most engaging fashion. Thank you for sharing your knowledge and experience. I think this was the first one, and the other one was second, but I read them out of order.
No problem.
But it's okay. All right, Dickson, here you go from Paul.
Okay, Paul writes, "Hi, guys. Just discovered the podcasts. Listening to 2 shows a day, working my way up. Absolutely love it. Love Dr. House, Parasite Rex, and Monsters Inside Me. While gathering information about parasites, I often see these so-called zappers, little devices that kill parasites. What are your thoughts on these, and the history of using electric devices in killing parasites? Greetings from Switzerland." You know, to be honest, I'm not familiar with them, so I can't honestly say what you mean.
Zappers?
I know zappers for flying insects, but I don't know any that would actually go down into your gut tract and zap things. I don't know about that.
Right.
I don't know about that one.
If you,
I need a larger explanation.
Paul, send a link.
Yeah, exactly.
Josh writes, I'm an auditor. To occupy my mind while I work, I started scouring iTunes for science-rich content. I saw your show TWiP and thought, why not? I think the first TWiP I listened to was 33. I was immediately hooked. I went home and subscribed to TWiP, TWIM, and TWiV that night.
Oh, wow.
Excellent. Currently, I'm listening to all of the TWiPs in a row before I start the other shows. I love the education. The ick factor is pretty high, but this show provides me with an excellent free education and helps me confront fears that were boogeymen in my mind by giving me real quantifiable behaviors and characteristics of many things I have had uninformed fears about.
You mean to tell me that you can eliminate fears by education? My goodness, how radical.
Amazing, huh?
I mean, if you don't stop research and you get the data, you could actually allay a lot of fears out there. Like, for instance, with H5N1, just to throw out.
Are you obsessed with that?
Not me.
I am very saddened that I didn't know who Dr. Despommier was prior to November of this year. In the spring of 2011, I was writing a business plan for my entrepreneurship class, and I advocated that my group do our project on vertical farming.
Wow.
We were unable to find any resource that would provide us with information on how to successfully integrate a vertical farming business into an urban area such as Phoenix, Arizona, where our fictitious business had to be. We had thought about potentially buying a tall structure like an empty condo tower or office building and converting it to a farm, or placing farms on rooftops, kind of like they do in Cuba. Sadly, we couldn't find any cost-to-benefit analysis, yield per acre data, or potential profit margins with which to build our fictitious business plan. We ended up using another idea of mine, recycled building material sales. I remain sad that we were unable to write a plan for vertical farming.
Me too.
Thank you for providing me with such a wealth of free information. Thank you for presenting it in such a content-rich way without spoon-feeding the listener. Please continue to do these shows. I am indifferent to frequency as long as I can continue to have access to your knowledge and experience. As long as we're alive, once we're gone, there's no more access.
None, but then you can re-listen to the other ones.
Would you like to read the last email, Dickson?
But I would like to answer a question before this. There's a guy in Chicago, his name is Benjamin Kennedy, and I was present at his thesis defense. He was an architecture student at the Illinois Institute of Technology, but presented his thesis in the office of the city of Chicago, to the sustainability office. And he had all of the things that you were missing in your business plan. So if you're still interested in revisiting that, you can get in touch with Benjamin Kennedy. He's listed in the directory, and he has a website for his architecture firm. So too bad you didn't. Too bad we didn't hook up. But it's never too late. Dear Dr. Racaniello and Dr. Despommier.
This is from Rachel.
From Rachel. I'm in the process of choosing a career, MD or PhD. I would love to follow in your footsteps and become a professor, researching parasites and leading the young minds to new discoveries. That's very noble. However, when talking with my parasitology teacher, it seemed like he was discouraging me due to the lack of funding and interest in the subject. I wanted to know your opinion, especially Dr. Despommier's. Do you think part of your success in your area of research came from the newness of the subject? Or is there still an opening for hopeful parasitologists? Thanks for all the work you both put in to the TWi series. I'm a huge fan. I am waiting anxiously for the next installment of TWiP. Well, here it is. It's not an either or. You could actually go for both if you're gifted enough to get accepted into an MD PhD program. Will there always be new things to discover about parasites, and therefore always a need for a supporting parasitology research? And the answer is, of course. We'll never know everything there is to know about these beasts. And in fact, we don't even know most of the important things we need to know about these beasts, as you've heard from these recent podcasts that we've had on various research aspects. So if I were advising you, I would not give you this gloom and doom view of the negativities associated with struggling to obtain funds to do research, but rather to tell you that there are many, many, many people out there who would gladly take you on after you finished your PhD as a postdoc, working in their laboratory. They're already funded, and you can start working on projects that they're interested in and maybe adopt them for your own, or learn all of their technologies and then adapt them to your own programs after you finish your postdoctoral experience. But I think there's multiple opportunities out there for a successful career in parasitic disease research, regardless of whether you have an MD or a PhD. And we see new people coming along all the time that contribute to the literature and make meaningful and gainful results that data can be then applied to many other systems.
Would you say that the field of parasitology is more robust than it was when you were in it?
it depends on the subject area of the parasitology that you're talking about. I think taxonomic areas are dead. You can work for museums, for instance, and work in the DNA lab and classify organisms based on their patterns of DNA expression and stuff like this. And that's got some usefulness, and museums do employ parasitologists as a result. But the current cutting-edge parasitology groups are focusing on the world health problems of malaria, leishmaniasis, lymphatic filariasis, and schistosomiasis. Those are the big issues that need to be solved, either with vaccines or new drugs or other approaches to interrupt the transmission cycles. And so, it depends on what you think constitutes research in parasites. But I think malaria has captured the largest share of NIH funding right now, and rightly so, because it affects so many people and kills so many each year. But let's assume that within the next 5 to 10 years, there will be new approaches to malaria, and maybe the reduction in malaria will be replaced by the next most prevalent diseases. And I think it's based on prevalence and medical histories and mortality rates as to whether the funding streams occur or not. So basic parasitology, I think they get the short shrift. You'd have to go to places like the National Science Foundation or some private funding agencies for that money, and it's very difficult to convince them that you're not working on a medical problem. So that part's true, but I don't think it's hopeless.
Well, if you'd like my 2 cents, Rachel.
I would.
Maybe she doesn't want my 2 cents, but
give it to her.
First, check out PLOS pathogens and PLOS neglected tropical diseases to see the state of the field. There's a lot of good work still going on.
Sure.
Funding should not discourage you. It always waxes and wanes. And as someone told me when I was a student, I was worried about funding. She said, there's always room for good people.
Bingo.
If you want to do it, just do it.
Bingo.
And don't worry about the money.
Follow your heart.
Yep. Do not worry about the money.
That's right.
All right. We got through all the emails. That'll do it for TWiP 36. 36, Dickson.
36.
Soon we'll be at 100.
Soon.
Can we make 100?
Absolutely.
Just kidding. You can find TWiP in iTunes at microbeworld.org/TWiP. And there is also an app over at Microbe World for playing the podcasts on your iPhone or Android device. If you have questions or comments, we love to get them. Send them to twip@twiv.tv. Dickson Despommier. Where can people find you?
Several places, actually. Fort Lee, New Jersey would be one of them. But they can find me on the internet listed under either verticalfarm.com, Tracaniello.org, or medicalecology.org.
The internet.
The internet.
You never knew that this was going to happen to you, did you?
I lived in fear.
Thank you, Dickson.
You're welcome, Vincent.
I appreciate your doing a TWiP at my beck and call.
I'd do it anytime you ask.
Thank you very much.
Anytime I can, I would do it.
I'm very good. I appreciate it.
Good.
I make millions, and I do appreciate you working for free.
That's okay. I'm retired. I don't need the money.
I'm Vincent Racaniello. You can find me at virology.ws, where I don't always write about H5N1.
No.
Just lately.
Yeah. We have a good reason.
You have been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.